So I want you to imagine something for a second. Um, Whenever you watch one of those, true crime documentaries, there's always that one inevitable scene.
Oh, yeah, the aftermath scene.
Right, exactly. The detectives roll up, the yellow tape is everywhere, and they're just staring at these chalk outlines.
Right.
And the whole investigation is, it's basically an exercise in total frustration.
Right.
Right. Because they are looking at debris. They're trying to reverse engineer this really dynamic, chaotic, moving sequence of events.
Yeah.
But they only have the tragic endpoint. And you, as the viewer sitting on your couch, you just want to yell at the TV like, if only you had a security cam.
Scroll the tape back.
Exactly. If you could just rewind the tape and watch what actually happened,
Yeah.
whole thing would be solved in 5 minutes.
Yeah, it's a fundamental limitation of observation. You are deducing an entire trajectory based completely on a corpse.
Right. And for the longest time, that exact frustration has been the defining reality in the world of biology. Because when scientists are trying to understand human cells, like how they react to a new drug or how they mutate, they haven't had a video camera.
No, not at all.
They've literally just had the crime scene.
Yeah.
But today, we are doing a deep dive into a really fascinating stack of research. It's centered around this incredibly provocative post by Paramendra Kumar Magad from May 2026.
Yeah, it's a great piece.
Right. And it details a company that claims they have finally built that video camera for living cells.
Yeah. And the reason this particular source material is so gripping isn't just, the sheer science fiction nature of the tech itself.
Right.
It's actually a story about a massive friction point. It's about this fundamental clash between a groundbreaking scientific capability and the entire financial ecosystem that's structurally supposed to fund it.
Yeah, it's wild.
We are looking at a literal rebellion against the traditional Silicon Valley playbook.
We really are. We're looking at a startup called PreciGenetics, and it's led by a founder named Parmita Mishra. And her central thesis is just audacious. I mean, she is actively claiming that her company is too big for VC.
Which is such a crazy thing to say in tech.
It is.
Yeah.
They are intentionally bypassing traditional venture capital, which, is a wild premise for a startup. Because usually if a founder says, oh, I don't want venture capital, your cynical assumption is
They just couldn't get the money.
Exactly. They couldn't get any term sheets. But the argument here is that the Silicon Valley VC engine is structurally incapable of supporting the scale of infrastructure they're building.
Yeah.
Okay, let's unpack this. Because to understand that financial rebellion, we first have to ground ourselves in the physical reality of what they're actually building.
We do. And the article uses this very stark phrase to describe the status quo of cellular biology right now.
Oh, right.
It says that currently, researchers have to, quote, kill the cell, stain it, sequence the corpse.
Which is just such a brutal description.
Yeah.
But it's accurate, right?
It's completely accurate.
My understanding is that historically, if you want to see specific proteins inside a cell, you can't just look at it through a normal glass lens.
No, it doesn't work that way.
You have to stop the biological process entirely. You lock the cell in place with chemicals, you inject it with fluorescent dye, so the parts you want to see light up.
Right, under a specialized microscope.
Exactly. But the second you do that, the cell's behavior is altered.
Or it's just outright dead.
Right. It's dead.
So you get highly specific data, sure, but the living, breathing biological entity you were actually trying to study is gone.
It's gone.
You just get your forensic snapshot.
Right. It's like trying to understand the strategy of a basketball game by only looking at still photographs.
Oh, that's a good way to put it.
Right. Like players suspended in midair, you see a player holding the ball, but you have no idea what the velocity of the pass is.
You don't see the defensive rotation.
Exactly. You miss the entire continuous flow of the game. So PreciGenetics is claiming to move us from those static photos to a live 4K broadcast.
Yeah. They call it cell cinema.
Cell cinema. I love that branding.
It's catchy. It's a platform that combines biophotonics and artificial intelligence to deliver real-time chemical movies of living cells in motion.
Without any labels or dyes, right?
Exactly. No destruction at all.
Okay, I actually need to pause here because the material mentions this involves, quote, label-free cell biophotonics.
Yeah, it's a mouthful.
It really is. And for anyone who doesn't have a PhD in physics, how does this actually work? How do you see inside a living cell without lighting it up with dye.
Well, what's fascinating here is that it all comes down to how light interacts with matter.
Okay.
So when you shine a highly calibrated laser into a bioreactor full of living cells, those cells don't just absorb the light, they scatter it.
They scatter it.
Yeah. As the light passes through the cell wall, the nucleus, the mitochondria, the beam, bends and refracts in incredibly complex ways.
Yeah.
And crucially, as the cell changes its chemical state, say it's starting to get sick or it's metabolizing a drug, its physical density changes slightly. Right, so that means the way it scatters the laser light changes continuously.
So you are basically bathing the cells in light and just reading the resulting optical chaos.
Essentially, yes.
But how do you make sense of that? If it's just a million scattered light waves, isn't that just noise?
Well, for decades it was just noise.
Really?
Yeah, and that is exactly why Parmita Mishra refers to this space as a barren land in her social media posts.
A barren land.
Yeah. She points out there are only a single-digit number of professors globally who even understand the deep mechanics of label-free cell biophotonics.
That's wild. Single digits.
Yeah. The hardware to shine the light and capture the scatter, that's existed in various forms. But human brains and even traditional software just cannot decode that chaotic wave pattern into a meaningful biological picture fast enough.
Ah, okay. So it's an AI play as much as a hardware play.
It's entirely both.
It sounds almost like building a Shazam for biology.
Oh, yes.
Yeah.
Exactly like that.
Like where Shazam listens to the chaotic audio waves in a crowded coffee shop and instantly identifies the underlying song. This AI is looking at the chaotic light scattering and instantly identifying the chemical song of the cell.
That is exactly the mechanism.
So it says like, that light pattern means this cell is exhausted, or that pattern means this cell is successfully fighting the cancer.
Right. It requires foundational artificial intelligence models, and they have to be trained on massive data sets of light scatter patterns mapped against known biological states.
Okay.
When you combine the photonic hardware with that AI layer, you suddenly have a non-destructive, continuous video feed of living biology.
You have cell cinema.
You have cell cinema.
And while that is a phenomenal scientific achievement, the source material quickly pivots to why this is a massive economic lever.
It really does.
Because we transition from the abstract physics of lasers into an incredibly urgent bottleneck in modern medicine.
Yes, the CAR-T crisis.
RT cell therapy.
Right. Chimeric antigen receptor T-cell therapy. It is arguably the most potent weapon we have ever developed against certain blood cancers.
It's a miracle treatment for some people.
It really is. But it is an autologous therapy.
Autologous, meaning it comes from the patient themselves.
Exactly. You have to extract the patient's own immune cells, genetically re-engineer them in a highly sterile lab facility so they grow custom receptors that hunt down that specific patient's cancer.
Wow.
And then you infuse those engineered cells back into the patient's bloodstream.
It's bespoke, artisanal medicine.
Highly artisanal.
And the material hits us with some absolutely staggering financial figures here.
The numbers are insane.
They really are.
Yeah.
The total cost of a single CAR-T infusion hovers around $500,000.
Yeah.
But the really shocking part is the breakdown. The text states that direct manufacturing, logistics, and quality control expenses, just the QC and manufacturing costs between $170,000 and $220,000 per patient.
Right.
I have to challenge this. How on earth does the manufacturing and quality control for a bag of cells cost the equivalent of a single-family home in the Midwest?
I know, it sounds impossible, but it's because right now that artisanal manufacturing is essentially flying blind.
Flying blind.
Yeah, you place these delicate, newly engineered cells into a bioreactor to multiply for 2 to 3 weeks.
Okay.
But because we only have those forensic snapshots we talked about, the only way to know if your multi-$100,000 batch of cells is actually viable and sterile is to physically open the system, extract a sample, and test it in a separate lab.
Oh, which introduces the risk of contamination every single time you take a sample.
Exactly. And the testing itself takes days or even weeks.
Oh, wow.
So imagine you are brewing this life-saving therapy. On day 4, the cells start becoming metabolically exhausted.
Okay.
But you don't know that. You don't find out until day 14 when the snapshot test comes back. By then, the entire batch is ruined. The patient doesn't get their medicine, and the manufacturer just burned hundreds of thousands of dollars in raw materials and highly specialized labor.
That's devastating. So by introducing cell cinema, by constantly reading the light scattering of the cells inside of the bioreactor, you eliminate that blind spot.
You completely remove the need for physical sampling.
Right.
The sensors watch the cells in real time. If the cells start showing signs of exhaustion on day 4, the AI flags it immediately.
That's huge.
The manufacturer can dynamically adjust the nutrients or the oxygen levels in the bioreactor to save the batch. You are drastically slashing the failure rate.
Mishra describes this in the text not just as a data generation machine, but as an urgent need in manufacturing. She literally puts urgent in all caps.
Because it is.
Right. Because cell therapy is currently the fastest growing segment in all biotech and pharma.
It has a massive compound annual growth rate.
But that growth is going to hit a brick wall if every single dose requires $250,000 of blind artisanal labor.
Exactly. They're trying to build the foundational infrastructure to digitize living systems at scale. They want to turn biology into an industrial, predictable process.
Which forces us to confront the central friction point of this entire analysis.
Right, the money.
Yes, the money. If PreciGenetics has the platform to solve a multi-billion-dollar bottleneck in the fastest-growing sector of medicine, why are they locking venture capital out of the room?
It's the big question.
Why not walk onto Sandhill Road, pitch the top-tier VC firms, and raise a massive Series A?
This raises an important question because it seems totally counterintuitive until you examine the actual mechanism of a venture capital fund.
Right. I mean, we constantly see headlines about venture capitalists dropping hundreds of millions of dollars into everything from generative AI startups to companies trying to build supersonic jets or, brain-computer interfaces.
Yeah, they take massive bets.
They take huge bets.
Yeah.
So the idea that VC is structurally incapable of funding cell cinema just feels wrong at first glance.
It does. But the author of this piece makes a very compelling case that VC is highly specialized for software. Okay.
Yeah.
If you are building a SaaS product, software as a service, your upfront capital requirements are relatively low.
Sure.
You write the code once, and the cost of duplicating and distributing that software to your second customer or your millionth customer is practically zero.
Right. The margins are astronomical.
Exactly. So a VC gives you $10 million. You use it to acquire users aggressively. You scale at breakneck speed. And within 5 to 7 years, the company goes public or gets acquired.
The VC gets their liquidity event.
Exactly. That 3 to 7 year liquidity clock is baked into the very structure of the fund they raised from their own limited partners.
Oh, I see.
But deep tech, especially physical infrastructure at the intersection of biophotonics, hardware manufacturing, and biology, it does not operate on a software clock.
Not at all. The development cycles are incredibly long.
Yeah. You are building complex physical lasers, dealing with supply chains, facing massive regulatory hurdles.
So the capital intensity just dwarfs a typical software startup.
It really does. And if you force a company like PreciGenetics into that traditional software-focused VC model, the author warns of 2 disastrous consequences.
Okay, what are they?
First, the founder dilution is brutal. Because you need so much capital over so many rounds just to build the physical prototype, the founders end up owning a tiny fraction of their own vision.
Wow. Yeah, that makes sense, because every round you raise, you give away a chunk of the company.
Exactly. And second, there is immense pressure for premature pivots.
Premature pivots.
Yeah. The VC's 5-year clock is ticking. The fund needs to show a return. So the board pressures the founder to abandon the massive, world-changing infrastructure project and pivot to a smaller, incremental product that can be monetized immediately.
Oh, like, hey, stop building the revolutionary camera. Just sell this slightly better microscope lens.
Exactly. You end up building a slightly better lab tool instead of the foundational network for the next era of biology.
Which is why the source material argues that PreciGenetics doesn't need equity-driven capital.
Right.
They need mission-driven capital.
Yes.
They need funders who aren't looking to flip the stock in 5 years, but who desperately need the technology itself to exist because it solves an existential threat to their own core business.
And to illustrate how to actually capture that kind of capital, the text brings up a fascinating historical blueprint.
Early SpaceX.
It argues that PreciGenetics needs strategic mega customers, much like NASA was for Elon Musk.
It is such a vital comparison, because today SpaceX is a ubiquitous giant. But in its early days, traditional Silicon Valley venture capitalists were largely terrified of it.
Oh, absolutely terrified. The capital required to build an orbital rocket from scratch was absurd.
And the risk of literal explosive failure was sky high.
Yeah. If SpaceX had relied solely on the standard VC drip feed, they likely would have starved to death.
But they survived because NASA stepped in through the CO test program, right?
Yes, commercial orbital transportation services.
Right. And NASA wasn't a venture firm looking for a 10x return on equity. NASA was a customer facing an existential problem.
The space shuttle was retiring.
Exactly.
Yeah.
The space shuttle was retiring, and they had no way to get cargo to the International Space Station.
They needed rockets.
They needed rockets. So they gave SpaceX massive milestone-based contracts, almost $400 million before the Falcon 9 even flew.
Which is the magic of the NASA-style anchor contract. It provides massive, non-dilutive capital.
Non-dilutive meaning you don't give up equity.
Right. SpaceX didn't have to give up board seats or ownership to get that money. They just had to deliver a working service.
Wow.
But more importantly, it provides supreme, unassailable validation.
Oh, validation. Because if NASA trusts you.
Exactly. If the most rigorous aerospace agency on the planet trusts your hardware, it completely de-risks the technology in the eyes of the broader commercial market.
It's the ultimate stamp of approval.
It really is.
Here's where it gets really interesting, though. The author's argument is that PreciGenetics needs to execute this exact same playbook.
Yeah.
Instead of chasing VCs, go find the NASAs of the biological world. Find the entities whose workflows are suffocating because they lack real-time cellular data and get them to fund the development via massive service contracts.
And the source doesn't just leave this as a theoretical exercise. It outlines a highly specific, very credible roadmap of 10 customer categories that could collectively provide over $100 million in non-dilutive funding.
Which is just a brilliant blueprint for bypassing the VC ecosystem entirely.
Let's actually walk through this roadmap because seeing the specific targets makes the scale of this vision feel real.
Yeah, let's do it. So the first major category is, unsurprisingly, the major pharma giants.
Right. Your Pfizers, Roches, Novartis, Mercks.
Exactly. And the use case here isn't just manufacturing, it's the very beginning of the pipeline. High-throughput drug screening.
Right. When a major pharmaceutical company is hunting for a new drug, they screen millions of chemical compounds against libraries of diseased cells to see what happens.
Okay.
But again, under the current forensic snapshot model, they administer a compound, wait days, and then test to see if the cell died or survived.
Right, which is so slow.
Incredibly slow.
The text notes that with cell cinema, they could watch the reaction in real time. They could predict the trajectory of the cell's response in hours instead of weeks.
Yeah.
If you were a pharma giant, compressing your drug discovery cycle by weeks or months across millions of compounds,
Yeah.
I mean, that translates to billions of dollars in saved R&D and faster time to market.
It also reduces late-stage failures, which is huge.
Oh, how so?
Well, right now, a drug might pass an early snapshot test, but fail years later in clinical trials because the snapshot missed a subtle, long-term toxic effect on the cell's metabolism.
Oh, I see.
A continuous chemical movie catches those subtle toxicities early before the company spends $1 billion putting it into human trials.
Wow, okay. That makes perfect sense. So pharma giants are an obvious customer.
Yep.
The next target on the list are the cell and gene therapy leaders, like Gilead, Kite, and Bristol-Myers Squibb.
Which brings us right back to our CAR-T discussion. They're the ones feeling the acute pain of those $200,000 quality control bills.
Exactly. They need this yesterday. And closely related to them is the third category, the CDMOs.
The contract development and manufacturing organizations.
Right. Companies like Lonza, Wookie, Catalent. I think the general public assumes that when Pfizer invents a drug, a giant factory with a Pfizer logo on it manufactures every single pill or vial.
Right, that's the common perception.
But often, they outsource the actual physical brewing and scaling to these massive CDMOs. They're like the shadow factories of global health.
They really are the industrial backbone of the industry.
And for them, the source text points out that biosensing acts as a service layer directly tied to something called PATE.
Process analytical technology.
Okay, P8. The text mentions it's a massive regulatory priority, but I mean, it just sounds like pure corporate jargon. What does process analytical technology actually look like in practice?
Okay, think about baking a highly complex cake.
Okay.
The old way, the non-pita way, is you mix the ingredients, throw it in the oven for an hour, take it out, cut a slice, and eat it.
Yeah.
If it's burned in the middle, you throw the whole cake away and start over. That's how a lot of legacy bioreactor manufacturing works.
They just wait till the end and hope for the best.
Right. Peat, on the other hand, is the equivalent of having a network of sophisticated thermometers and moisture sensors embedded inside the cake while it bakes.
Oh, wow.
And those sensors are feeding data back to the oven to dynamically adjust the temperature second by second, so it literally cannot burn.
It's moving quality control from the end of the line directly onto the manufacturing line itself.
Precisely. And regulators are desperate for CDMOs to adopt peat.
Really?
Because it guarantees a vastly safer, more consistent product. PreciGenetics offers the ultimate PAT solution for living cells. It's not just a thermometer, it's a real-time chemical analyzer.
So the CDMOs are a massive market. But wait, if fixing corporate profit margins for pharma and CDMOs was the whole story, it would still be a massive business.
Well, a multi-billion-dollar business.
But the roadmap takes a hard pivot away from the private sector here.
It does.
It suggests the biggest buyers for this infrastructure are entities whose mandate isn't profit at all, but national security and public health.
We are talking about the United States government.
Yes. The U.S. government has a scale of purchasing power that just dwarfs almost any private corporation.
Totally. The text specifically highlights biomedical research agencies like the NIH, BARDA, and heavily emphasizes ARPH.
ARPH, the Advanced Research Projects Agency for Health.
Right.
It's essentially the healthcare clone of DARPA, right? The defense agency that famously funded the creation of the internet and GPS.
Exactly the same model. ARPH doesn't want to fund a slightly better aspirin. They exist exclusively to fund massive, high-risk, paradigm-shifting leaps in medical capability that are too structurally daunting for the private sector.
And building the fundamental hardware-software platform to digitize living biology is the exact definition of an ARPH mandate.
It is. They can drop a $50 million or a $100 million grant to accelerate the development, not because they want equity, but because the U.S. healthcare system fundamentally needs the capability to exist.
And speaking of DARPA, they are explicitly on this mega customer hit list too, along with DTRA, the Defense Threat Reduction Agency.
Yes, the national security angle is huge here.
The text lists applications for biothreat detection, rapid countermeasure development, and soldier health monitoring.
Consider the biodefense posture of a nation.
Yeah.
If an unknown pathogen or a novel engineered bioweapon appears, the speed at which you can understand its mechanism of action dictates how many lives you save.
Obviously.
If your national labs are relying on forensic snapshots, you are always days behind the pathogen.
Right, because you have to keep killing the cells to see what the virus is doing.
Exactly. But if you have cell cinema, you introduce the pathogen to human cells in a secure lab and watch exactly how it breaches the cell wall in real time.
Oh, wow.
You test countermeasures and see instantly if they halt the breach. It turns biodefense from a reactive, slow-motion crawl into a real-time tactical response.
That is an incredible visual. It's not just a medical tool anymore. It's a literal national security asset.
It is.
And then there's the FDA. The text suggests collaborative programs with regulatory bodies. Now, this one confused me at first.
Really? Why?
Well, why is a startup treating the FDA like a customer? I mean, usually founders view regulators as a massive obstacle to overcome, not a strategic partner.
Yeah.
Are regulators just tired of taking pharmaceutical companies at their word when it comes to safety data?
Well, it's about solving the regulator's core information deficit.
Information deficit.
Yeah. When the FDA reviews a new cell therapy, they are entirely dependent on the snapshot data the manufacturer provides. But the FDA's mandate is absolute safety.
Right.
If they collaborate with a company like PreciGenetics to validate this new continuous monitoring technology, the FDA can eventually shift the regulatory standards.
Oh, like raising the bar.
Exactly. They can say to the industry, look, we know continuous real-time safety monitoring is physically possible now. Therefore, we are no longer accepting batch-tested snapshots. You must integrate PAT to get approved.
Wow. By adopting the tech early, the FDA raises the bar for the entire industry. And PreciGenetics positions themselves as the gold standard that the regulators themselves helped validate.
It is a masterclass in aligning public health incentives with deep tech development.
That is brilliant strategic positioning, but the roadmap doesn't stop at human health either.
No, it goes much wider.
Part 3 of this customer landscape pushes the boundaries into entirely different industries. It lists agri-biotech and synthetic biology. Companies like Corteva or Ginkgo Bioworks.
Yeah, we often forget that synthetic biology is quietly rebuilding our physical world.
What do you mean?
Well, companies are engineering yeast and microbial cells to consume plant matter and secrete everything from sustainable aviation fuel to biodegradable plastics to advanced fertilizers.
Okay, sure.
But tuning those microbial factories to run efficiently at an industrial scale is painfully difficult.
Because they are currently treating the yeast cells like black boxes.
Exactly. You feed them sugar, wait a week, and see how much plastic they made.
Right.
If PreciGenetics can put their optical sensors on an industrial fermentation tank, you can watch the microbial metabolism in real time and optimize the environment instantly.
So you maximize the yield of sustainable chemicals.
Exactly. You make the whole process efficient.
And then the text loops in big tech. Google DeepMind, AWS Health, Microsoft Research.
Yeah. This is where the AI half of the platform becomes the primary product.
The suggestion is to co-build foundational models trained on cell cinema data streams, which, I mean, if we look at the AI boom happening right now with large language models like ChatGPT.
Right. They're incredibly powerful because they were trained on basically the entire written text of the internet.
Right, but biological AI is bottlenecked because the data doesn't exist.
Exactly.
You can't scrape the internet for real-time, non-destructive chemical movies of human cells reacting to drugs.
It simply hasn't been recorded yet. PreciGenetics isn't just building a tool, they are building the camera that will generate the training data for the first true biological foundational model.
That is massive.
The massive tech conglomerates would pay astronomical sums for exclusive access to that fire hose of novel, high-definition cellular data to train their own healthcare AI systems.
Which brings us to the most surprising and frankly fascinating target on the list.
The German connection.
The German connection. Parmita Mishra specifically called out European hardware and photonics companies on her social media.
Yes, she did.
She noted that the only companies initially showing deep interest were in Germany, and she diagnosed this by saying they have a lot of hardware tech debt.
If we connect this to the bigger picture, this is a profound insight into the global engineering landscape.
How so?
If you look at the history of precision optics, Germany is the undisputed sovereign. Companies like Carl Zeiss or Leica have spent over a century perfecting the physical manipulation of light.
Okay.
They understand lenses, lasers, and physical optics at a level that Silicon Valley software engineers just cannot replicate.
Right. They are hardware masters.
Exactly. They see the physical biophotonics required for cell cinema, and they instantly respect the monumental engineering challenge.
They get the hardware. But what does she mean by hardware tech debt?
Well, legacy hardware companies are fundamentally structured around physical perfection. But we have entered an era where physical perfection is no longer enough. The value is in the software layer that interprets the physical data. German optics firms often struggle to transition into modern, cloud-based artificial intelligence. They can build a microscope that captures petabytes of chaotic light scatter, but they lack the cutting-edge machine learning talent to decode that scatter into a readable video.
That is their tech debt.
That is their tech debt.
So PreciGenetics becomes the perfect symbiotic bridge. By partnering with legacy German optics, PreciGenetics effectively gets access to a century of the world's best physical engineering and supply chains for the hardware.
Yes.
And in return, they provide the state-of-the-art AI software models that the Germans desperately need to modernize their equipment.
It bypasses years of hardware prototyping. It is a highly pragmatic, globally aware strategy that completely sidesteps the Silicon Valley venture capital echo chamber.
And that pragmatic strategy perfectly echoes the founder's overarching philosophy. The text highlights a quote from Mishra where she states, "Our IP is translational. We aren't early, we are just in time."
Just in time is such a powerful framing. It acknowledges that 5 years ago, the AI models weren't fast enough to decode the light scatter.
Right.
And 10 years ago, the cell therapy manufacturing crisis wasn't acute enough to force the industry to change. The technology, the computing power, and the economic desperation have all converged at this exact moment.
She's intensely aware of the cultural divide she is straddling, too. She mentions creating her social media presence specifically to, quote, bridge the unsexy biotech pharma world in Silicon Valley.
Yeah, she knows that Silicon Valley VCs want frictionless, viral software that scales overnight.
And she knows that traditional pharma moves at a glacial, highly regulated pace. She is trying to translate the monumental importance of building hard physical biological infrastructure into a language of speed and scale.
She even states, "If I had 1B in funding today, we would take over so fast, but we will build our way to it." It's an acknowledgement that the traditional path is closed, but the destination remains unchanged. The era of observing living biology continuously is coming, and it will be funded by the customers who are desperate for the data, not by investors speculating on equity.
As we wrap up this massive exploration, I want to pull all of this esoteric science and financial strategy back down to earth.
Sure.
If you are listening to this and you don't run a cell therapy lab, you aren't an ARPAH program manager, and you aren't a venture capitalist, why does the funding strategy of this specific deep tech startup matter to your life?
Because the friction we discussed today dictates the trajectory of human health.
Okay.
The way we fund infrastructure directly impacts your body. If you or a family member ever require personalized cancer therapy, the reason it currently costs $500,000 and requires a massive fundraising campaign to afford is because the manufacturing line is flying blind.
If the underlying infrastructure remains trapped in the forensic snapshot era, life-saving medicine remains an artisanal luxury.
Exactly. The speed at which the next pandemic vaccine is developed, the safety of the drugs you take, the viability of sustainable biochemicals that could replace plastics, all of these depend on our ability to observe and understand living cells in real time.
Yeah.
If our brightest scientific minds are forced by the venture capital system to abandon massive foundational projects and build small, incremental software tools just to secure funding, we collectively delay the future of medicine.
PreciGenetics is fighting a structural battle to build the tools that rewrite how human health is managed.
They are rewriting the financial playbook to force the scientific future into existence.
We've covered an incredible amount of ground today. We explored the physics of label-free biophotonics and how AI acts as a decoder ring to turn scattered light into cell cinema.
Yeah.
We looked at the agonizing bottlenecks of CAR-T manufacturing. We analyzed the structural timeline mismatch that makes traditional VC hostile to deep tech. And we walked through a $100 million roadmap of mega customers, from DARPA to legacy German optics firms, willing to fund the revolution via service contracts.
It is a blueprint for how hard tech can survive and scale outside the traditional bubble.
Which leaves a truly provocative question hanging in the air.
What's that?
If a deep tech startup can successfully bypass Sand Hill Road, if they can build the foundational biological infrastructure of the 21st century by going straight to the government agencies and pharmaceutical giants who actually need it, what is the long-term relevance of Silicon Valley venture capital?
Oh, wow.
Right. If the traditional allocators of capital cannot adapt to the capital intensity and timelines of physical science, will they be left fighting over dating apps and SaaS enterprise software while the actual future is built without them?
It's an existential question for the investment class. Capital flows to where it is most effectively deployed, and right now the mega customers might be the most effective VCs in the world.
Something to think about the next time you hear about a massive Series A funding round for a software tool. Or the next time you look at a static photograph and wonder what happened the moment after the flash went off. Because the video feed for Living Biology is finally coming online. Thank you so much for joining us on this deep dive.